Yesterday we started with section 4, chapter 8, which is transcription. We said that uh, Crick, the scientist Crick, he suggested the central dogma. And we said, what is the central dogma? The central dogma, which means that the central dogma states the information inside the cell flows in one direction. In one direction. What that means? يعني احنا we said in our chapter, in this chapter, we are talking about something happened from DNA to protein. How this process happened? How the instructions from DNA to protein processed inside the cell. Okay? So he states this or defines the central dogma, which means how the information flows in one direction. من الدي ان اي اللي بتعطي instruction for the cell to RNA to protein. يعني for the proteins to be made inside your cell, they must the information must flow in this direction. The DNA هي اللي بتعطي instructions. The RNA take these instructions as a message, and outside the nucleus the protein will be made. وإحنا قلنا إن the main one from the important functions of the cell اللي هو making proteins. ليش؟ إن البروتينز they are very active in our body, and many enzymes in our body are proteins. وهدول الانزيمز they are needed by our body in many functions. يعني بالdigestion بنحتاجهم, in breaking food, in taking out waste, in everything we need these enzymes. فعشان هيك the main important function of the cell is making protein. To make a protein دائما instruction comes from the DNA لأن ال DNA have the genes. If your body needs a certain protein, the DNA will tell your cell to make this protein. فهي بتعطي instruction for the cell to make which protein. تطلع الانستراكشن هذا simply الانستراكشنز بيكونوا على الدي ان اي inside the DNA genes. DNA cannot leave the nucleus. الدي ان اي دائما مكانها inside the nucleus. DNA just give the instructions. How? There is a messenger RNA, which is a molecule that links between the DNA and the cytoplasm. هي أنا تخيلوا هاي السل. Okay? Here is the nucleus, and outside is the cytoplasm. Baking proteins doesn't happen inside the nucleus. It happens when outside. احنا تعلمنا انه بيصير بالرايبوزومز بيكونوا outside بالcytoplasm. الرايبوزومز هم the main factories for proteins. Okay? To make these proteins, DNA ما بتطلع برا النيوكليوس. It will stay inside. تعطي instructions for the cell to make it. How these instructions will move or will flow to the protein by a messenger RNA. هو molecule خصوصي زي بياخد المسج and take it out of the nucleus to the cytoplasm, and the protein will be made out in the cytoplasm. فعشان هيك هو عمل هذه السنترال دوغما that state إنه الانفورميشن دائما flows in the same direction من ال DNA which give the instruction. ال RNA هذا المسنجر which links between the DNA and the protein synthesis outside in the cytoplasm. تاخذ هاي الانستراكشن بالمسج and out in the cytoplasm the protein will be made. هذا ال flowing of the central dogma. هيك ال information will flow. Okay? احنا we said so central dogma includes three processes. أول process اللي هي replication for DNA, and we said this happens only once during the life cycle of the cell cycle of the cell. And the other process is transcription of RNA and translation. احنا our lesson is about the transcription. Okay? فدائما keep in your mind RNA هو molecule which is a nucleic acid molecule which links between the DNA inside the nucleus and the protein outside in the cytoplasm. تمام؟ هلا let's talk a little bit about the RNA. إحنا we learned that nucleic acids are one of the 5 carbon-based molecules. وقلنا هدول nucleic acids are polymers that made of a nucleotide. عندنا two types، قلنا DNA وعندنا RNA. هلا what is the difference between the DNA وال RNA؟ بالستراكشر. شوفوا ايش بالستراكشر. هلا ال DNA هو deoxyribonucleic acid. أما ال RNA stands for ribonucleic acid. Okay? The difference is الشجر بيكون عنده هو رايبوز شجر. في ال DNA اسمه deoxyriboz. تمام؟ فالرايبوز differs in the structure. في عنده extra oxygen atom here in the sugar. فهذا اسمه رايبوز شجر. ال RNA عنده رايبوز شجر، ال DNA عنده deoxyriboz شجر. تمام؟ في there is another thing in the base pairing. احنا تعلمنا بالدي ان اي عندي 4 bases اللي هم thymine, adenine, guanine, cytosine. But in RNA instead of thymine there is something called a base called uracil. يعني الدي ان اي فيها thymine, adenine, guanine, cytosine. اما ال RNA فيها adenine, uracil, cytosine and guanine. Okay? Also, there is another difference إنه عندي أنا ال RNA is a single stranded structure not double as you see here. شايفين أول شيء هاي ال DNA وهاي ال RNA they are the same. What is the difference؟ إنه ال DNA عنده deoxyriboz sugar وعنده هون thymine as a base مع cytosine, adenine, guanine. وال RNA عنده uracil instead of thymine. والsugar هو ribose. هلا as you see here the DNA is double stranded molecule ما بيكون one strand double stranded. ال RNA only one strand. Okay? Is it clear the difference between these two molecules?
Yes.
All right. So now هلا during the transcription there are 3 types of RNA اللي هو ribonucleic acid will be made. تمام؟ each one of these 3 types has a specific function. هلا احنا before we talk about these 3 types I would like first to show you this figure. هلا what is happening before talking about this figure I would like you to watch this nice video. It's very helpful. Okay, just let's start here.
In order for our bodies to function, we need to supply them with a variety of nutrients we get from our diet. In order for our bodies to function, we need to supply them with a variety of nutrients we get from our diet. Our bodies cannot use the food.
Grammarly can help you write quickly and confidently, so you never have to slow down at work. Instant correction
As it is when it enters our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients.
شايفين into when your body is functioning, okay, many processes are happening inside your body. One of these processes is digestion of food. Okay, هلا this is the let's say this is the digestive system lining. في عندي هون أنا different enzymes to break down the food. The food cannot be break down alone. They need enzymes to break it down. So here in the process of digestion, many enzymes needed to make this process. وهدول الانزيمز are proteins.
Our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA.
So my body needs this type of enzyme. So your cells should make this protein, should make this enzyme to supply your digestive system with these enzymes so they can work and break down your food. فهدول instructions to make this protein موجودين وين؟ in the DNA. Gene طبعا.
DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Gene
هلا let's say أنا I need an enzyme amylase خلينا نحكي amylase enzyme for my body. هلا هذا ال DNA have many genes. Each gene codes for a certain trait or for a certain protein, making protein. Okay? كل جين كل جزء من هذا ال DNA is coding, is a code to make a certain protein. تمام؟ ف let's say إنه هذا البروتين that we need is in this part of DNA. Okay? And we need to copy the instruction from this part of the DNA to make this protein. So it will start by promoter. في عندي promoter come here to tell where to start. Basically promoter on this part and the another who promoter which where to start and terminator region where to end. I just need only this part to be copied.
Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a
في عندي شغلة تعرفوها الريبليكيشن والترانسكريبشن هذول two processes must be regulated. اوكي شو يعني لازم يكون في عندي regulation إنهم. They shouldn't happen like this. Only they are regulated by some sequences على ال DNA where to start, where to stop. So for the body to make the protein that is wanted in the time you need it. Okay?
protein in a process called gene expression. For these genes, expression can be divided into is used to synthesize a protein that contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these
يعني شو يعني جين اكسبريشن؟ شو يعني جين اكسبريشن؟ يعني أنا عندي هون جين. هدول الجينز عبارة عن إيش؟ عن أدينين، ثايمين، جوانين، سيتوسين اللي هم sequence of bases. أوكي؟ الجينز عبارة عن إيش؟ عن sequence of bases. هدول البيزز هم codes. أوكي؟ هذا الكود must be يعني يصير له copying. أوكي؟ مين بياخذ هذا الكوبي؟ بياخذ ال RNA. بياخذ هاي المسج as a copy من هدول السيكونسز to make this protein.
These genes, expression can be divided into two processes, transcription and translation.
Okay. Expression means يعني how this genes هذا what that means how it will be expressed. Okay. تعطيك الexpression يعني what that sequence of these bases means which protein to do. To express the genes to express these genes عشان تتفسر هاي الجينز في عندي two processes must happen هي transcription و translation. احنا رح نحكي عن transcription اليوم not the translation.
In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase.
هلا احنا نتذكر انه بالريبليكيشن DNA replication الانزيم that build up a new strand كان اسمه DNA polymerase. Okay? But in transcription, the enzyme which is responsible to build an RNA اسمه RNA polymerase. طبعا this enzyme does not work alone. It works with the help of other proteins. Okay, بيكون complex مع بعض. Okay, and they assemble together to make this process. What will happen now? Look,
This process occurs in 3 stages: initiation, elongation, and termination.
هدول الستيجز I don't need it. I don't need you to know this. خلاص ما بدي تفاصيل بس بدنا نعرف البروسيس.
During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind.
هذا البروموتر recognition to tell the RNA polymerase where to start. تيجي هون عندي promoter أنا. يلا start from this segment.
This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open.
هلا احنا we need the instruction at this segment where the enzyme bonded. Okay, this will cause this segment only of the DNA to unzip. It will open just from this part because I need the instruction at this part only, which code for a certain gene. تمام? And the unzipping will happen here. Okay.
Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides.
تمام. هون I want to show you something. Just a minute. Okay.
By the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up,
Okay. Now here, what is the function of the RNA polymerase? زي الفانكشن تبع DNA polymerase. Okay? عشان يعمل new strand. But in this case, it's making a strand of RNA, not DNA. mRNA, messenger RNA. Okay? As you see here, we have 3 nucleotides in the nucleus. هدول 3 nucleotides will be joined to the exposed bases. هدول البيزز which are exposed in this base pairing rule. بالDNA كان الأدينين مع الثايمين، بالRNA بيكون عندي الأدينين مع اليوراسيل، لأنه الRNA ما في ثايمين، في يوراسيل instead of ثايمين. تمام؟ فبصير عندي أنا محل ما في أدينين بيجي يوراسيل to build the sorry محل ما في ثايمين بيجي يوراسيل to build the RNA. هذا هون as you see here is the RNA molecule that is forming. هذا ال RNA molecule عم يعمل building للنيوكليوتايد to get the message من ال DNA. أخذ المسج من ال DNA.
the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other.
شايفين؟ خلص. هلا عندي أنا ال RNA polymerase إجا للترمينيتر ال end of the gene which we need to transcribe, and they are all will be detached بيروح الانزيم والRNA هلا هون strand will have the message. he got the message من وين؟ من ال DNA. هذا هو الترانسكريبشن. كأنه الترانسكريبشن هو ايش؟ converting هدول الانستراكشنز اللي في DNA to a message. أخذوا الانستراكشن to a message. هذا ال RNA now will go out from the nucleus to the cytoplasm to make the protein.
The strand of messenger RNA that is made during transcription includes regions called exons.
هلا before this process finished هذا ال RNA لسه not ready. في عندي processing must happen لأنه ال RNA بيكون فيها some exons و introns، فيها part coding for the protein وفي عندي part not coding يعني زيادة. ف what will happen to this RNA؟ قبل ما يطلع على النيوكليوس لازم يصير عندي processing له. What is the meaning of processing? يعني to make it ready for the protein synthesis. What will happen during this processes?
that code for a protein, and non-coding sections called introns.
إذا الإكسترونز هم الكودينج اللي هم عليهم الكود وفيها part introns وفيها بهدول they don't have code, they should be removed from the RNA.
In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added.
بيحطوا له بيزيد عليه cap وtail حنحكي عنه هذا in details in the next lesson. We'll add for it a cap and tail. Okay, and
This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature
هلا what is important to know here إنه أنا عندي صار removing of the introns and زي ما بنقول لزقوهم مع بعض they attach the other parts together. Now RNA is ready to go out to the cytoplasm where the protein should be synthesized.
mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation.
شايفين؟ هلا هون هذا ال RNA حامل المسج عشان هيك اسمه messenger RNA. تمام؟ هذا المسنجر RNA هلا will take this message to the cytoplasm وراح يصير عندي الترانسليشن. شو هي الترانسليشن؟ عم نفسر هاي المسج. We want to translate it to make the protein. طيب مين بتذكر what is the factory for the protein؟ مين هو الأورجانال that make the protein in the cell؟ مين؟ What is the main organelle in the cell that makes the protein? Who remembered؟ احنا قلنا في عندنا many organelles. عندي endoplasmic reticulum، عندي Golgi apparatus، عندي ribosome، central vacuole. What is the main function? Which one of these has the main function to make the protein? Yes, يا مراد.
Is it the Golgi apparatus?
No. الجولجي apparatus بيعمل packaging للبروتين packing وموديفيكيشن. مين اللي بيعمل؟
Endoplasmic reticulum.
لا الاندوبلازميك ريتيكولم هو السايت فور رايبوسومز وين الرايبوسومز بتتجمعوا والرايبوسومز هم الاورجانيز that are the factories of protein هم اللي بيعملوا بروتين. تمام؟ هلا هذا الار ان اي هلا هذا درس جديد انا ما راح اشرح عنه اليوم بس هيك افرجيكم بس هنت عنه عشان تفهموا كل الفكره مع بعض. هلا هذا الام ار ان اي will go out having the messenger. تمام؟ المسج هاي المسج must be translated outside the nucleus اللي هو in the cytoplasm to make the protein. عشان to be translated هذا ال RNA راح يمسك مع الرايبوزوم اللي هو الفاكتوري of protein. ياخذ هذا الرايبوزوم المسج and to make the protein according to this message.
How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called
هلا this will talk about it later. هلا هذا الترانسليشن هذا ال RNA ال mRNA have the message. الرايبوسوم will bind to this mRNA and get the message and translate it to which protein it should make. تمام؟ هلا let's go back to our PowerPoint. هلا guys, do you understand the idea? Guys, do you understand the idea?
Yes.
Okay.
يعني
Let's go back now to our PowerPoint slide and review what we said. Yeah. First of all, who can tell me briefly what is transcription? Yeah. What is transcription? يلا who want to yes Lujane. What is transcription, Lujain?
We could say that it's the change of information from DNA to RNA.
Yes, excellent. Converting the instructions اللي هم
Teacher,
الانستراكشن
can I say?
sorry,
Can I explain the meaning as well?
just 1 minute. يعني
Okay.
Lujain, you answered correctly, but you have to know that what we are converting in the transcription, we are converting the instructions coded in the DNA, okay, to a message, right?
Yes.
Okay, Lujain, yes. Abdullah, would you like to add something?
I was gonna say it's like the process of copying like segments of the DNA into RNA.
Excellent, excellent. So what are these segments?
Just as you said, teacher, like
Bit
can
of
encode
what?
protein.
Bit of what؟ لا هدول السيجمنتس in the DNA they are genes صح؟ طيب هدول
Yes.
الكود made of what؟ Yes مراد.
الفور bases
يعني
الأدينين
4 bases. الفور nitrogen.
يعني
Excellent. هدول البيزز اللي هم الادينين والثايمين والجوانين والترايتوسين هم coding. هدول coding. They will be transcribed, converted as a message on the mRNA. تمام. هلا I want to go back to our slide. Okay. Again, what is the difference between mRNA or RNA and DNA? The structure. What are the 3 differences between these 2 molecules? I want someone else. Baha, can you answer please? Yusra.
مس عيد السؤال.
What are the 3 differences between DNA and RNA?
Miss, can I answer?
Miss, I know.
يلا عبد
Yes,
الرحمن.
miss.
Give me one وسديل one. يلا عبد الرحمن.
Now, do you
Miss,
call me?
the DNA has deoxyribose sugar.
Deoxyribose sugar, excellent. And RNA?
Has ribose sugar.
Ribose sugar, excellent. Sadil, what is the second difference?
The second difference is it has the DNA has AGC, which is adenine, guanine, cytosine, or thymine. The other one has adenine, guanine, cytosine, or U. What does the U stand for?
Uracil, yes. Instead of thymine, uracil. Excellent. There is a third difference. What is it? Who can tell me?
Can I say something? Does it have to do?
Let's give chance for someone else. Yes, Lujain.
DNA is double-stranded while RNA is single-stranded.
Excellent. Bravo. أيوة. DNA is double-stranded helix while RNA is single-strand. Thank you very much. So, we said this process happens in a segment of DNA. So the unzipping of DNA happens in a certain gene, which codes for the protein synthesis. And what's the name of the enzyme needed for this transcription process? What's the name of this enzyme, guys? يلا يا سبيل.
إيش حكيتي مس؟
What is the name of the enzyme needed for this process?
Enzyme needed for this process. The transcription.
The transcription, yes.
The RNA polymerases.
Yes. RNA polymerase and other proteins that help the enzyme catalyze this process. All right. طيب. All right. As you see here, highly nucleotides, free nucleotides will be added to the open-stranded DNA, and as a result, RNA molecule is forming here. هذا ال RNA get the message for which protein is needed by the body. تمام؟ طيب, I would ask you a question now. Do you think transcription happens one or more in the cell? What do you think? Yes. Yes, يا مراد.
I think transcription happens more than one time and
Yes. Millions of times. Whenever your body need a certain protein, so transcription will happen to take the instruction from DNA and this will be translated later to make this type of protein. But the transcription is different from replication in this point. احنا قلنا بالreplication it happens once during the cell cycle, while in transcription is needed many times. يعني ممكن يكون عندي transcription أنا at the same time على DNA in different segments. Alright? هلا the last thing I would like to talk about اللي هو we have 3 types of RNA. هلا during the transcription there are 3 types of RNA are made inside the nucleus. The first one which we talk about اللي هو mRNA, which is responsible to carry the messages from the DNA out to the cytoplasm. Alright? Now, after this RNA take the message, there is another RNA will be needed in the translation اللي هو we call it ribosomal. Why we call it ribosomal? Because it will be a part of the ribosome. احنا قلنا انه الريبوزوم هم factories of proteins, right? فعندي انا كمان RNA اسمه ribosomal RNA will be part of the ribosome. And the last one is transfer RNA, which brings amino acids from the cytoplasm to ribosome. The last one, you will understand it more in the next lesson. Okay? Hella, did you get the ideas, guys? All the ideas about transcription? Okay.
Miss, yes.
Any question, guys?
Miss, no.
عبد الرحمن.
مش حاسس انت مش كنت تسأل يعني replication it makes more than million times.
Replication لا. احنا قلنا replication happens once during the cell cycle. لأنه each cycle should be divided once. صح؟
No.
No. عبد الرحمن. عبد الرحمن.
Hello.
Hello.
سامع.
Can you hear me? احنا we said that replication should happen once during the cell cycle. صح؟ صح؟ ليش؟ Why replication happen once؟ لأنه cell will divide once in its cell cycle. So replication needed one time. But the transcription is a process to make proteins, to take messages for making proteins. And we need these proteins every single minute. We need these proteins for our body to function properly and efficiently. Do you understand, Abdul Rahman? Yeah, miss, stay with me.
Yes.
Sadie, do you have any questions here?
لأنه بس أنت صوتك شوي بيقطع عندي.
Sadie, do you have any question?
لا يا مس لا كله تمام.
Okay, حبايبي. No one have any question. I have to leave now. If you have any question, please, you can ask. Abdul Rahman?
Okay, what is the difference between rRNA or tRNA?
اللي هو قصدك ال types of RNA?
Yes, there is rRNA.
شوف حبيبي هلا احنا the first one حكينا عنه اللي هو mRNA اللي هو المسنجر that carry the message. Okay, in the transcription صح؟ هدول rRNA وال tRNA هدول they are used in the translation اللي هو in the next lesson. تمام؟
Okay.
Each one of these types have a certain function for making proteins. تمام؟ هلا انت you understand what is the major function of mRNA?
Yes, miss, of course.
الريبوزومال والترانسفير خلوهم للدرس الجديد لانه هدول you will understand it more when we start the new lesson. Okay يلا take care guys.
مس
الله يعافيكم حبايبي. يلا take care. باي باي.